Hey guys, welcome back. Last time we talked about Mendel's laws, and we left off with this beyond simple dominance slide, so we're going to go ahead and wrap up this chapter here. Mendel focused on traits that had clear dominance and clear recessive alleles, but the expression for genes in different species, including humans, are not always that straightforward and simple. Our first example of an exception to this rule is called codominance. Codominance is where two non-identical alleles of a gene are both fully expressed in a heterozygote. Think not blended together, but independent and completely expressed. So there is really not a dominant, nor is there a recessive. This can occur in some multiple allele systems. We have lots of multiple allele systems. They're any gene where you have three or more alleles, and our most common one that we're familiar with is called the ABO blood typing in humans. So here's what this looks like. If you have A blood, you might have got an A allele from mom and an A allele from dad. They both code for a surface antigen that's A, and it gives you A blood. Or you might have got an A allele from mom and an O allele for dad. Now, O alleles don't code for a surface antigen, and so they're fully expressed. It just happens that their full expression is to not make a product on the surface of the cell, and so you still have A blood. Here's your best example of codominance. Mom gives you A, dad gives you B. Both of these alleles are expressed completely, and you have AB blood. The A expresses the A antigen on the surface of the red blood cell. The B expresses the B antigen on the surface of the red blood cell, and you have both of these gene products fully expressed, not blended, independent of each other, both making a gene product in your body. Big B, mom and dad both give you B, you're going to have B blood. Mom gives you B, dad gives you O, you're going to have B blood. And the only way you're going to have O blood is if mom gives you O and dad gives you O. And then you will be expressing no surface antigens. And so AB blood is our great example of codominance in these systems. The other option is incomplete dominance. Incomplete dominance, an allele is not fully dominant. And so this incomplete dominance, you can think of, it looks like there's blending in these traits. So the heterozygote phenotype is somewhere between the two with a 1 to 2 to 1 phenotype ratio. And I'll show you what this looks like. In your snapdragons, big R is red, little r is white, just like Mendel's peas. But big R is pink. Simple dominance, if this had been peas, would be red is dominant, white. So it'd be red, white. But in snapdragons, there's incomplete dominance. So you get red, pink, white. The good news is we can use those same laws, independent assortment and the law of segregation, to make our Punnett squares, do these experiments the same exact way. All those laws still apply. Just our phenotypes are a little bit different. So here's your homozygous parent. Big R, it's red. Here's your homozygous parent. Little r, it's white. We cross the P generation and we get an F1 generation. F1 generation is big R, and it is pink. So now we take this to an F2 generation. We cross two of these pink plants and we get this. Big R from pink parent. Big R from pink parent. We carry them across. Big R, red flower. Big R, pink flower. Big R, pink flower. Little r, white flower. And so your ratio becomes 1 to 2 to 1. 1 red, 2 pink, 1 white. Different ratio, different outcome in the phenotype for our heterozygotes, but all the laws still apply. So we haven't truly shattered all of our other things we had learned, like I had kind of teased you about a little bit. We can still use that stuff, that's nice. Okay, here's a fun concept. And this one we're getting really close to reality. Really close to reality. This is called epistasis. Two or more gene products influence a trait. So one trait might be dependent upon multiple genes And you see a lot of this in humans too. Typically, one gene product can suppress the effect of another. And one good example, it's pretty well studied, is our coat color in dogs. And so we have a big B and a little b, and those are our coat colors of the dog, black or brown. We have big E and little e, and that is the melanin color. How much melanin are they going to express? More of a brown melanin or more of a red melanin? And so the big E is brown, the little e is red, and your coats can be black or brown. Now, your dog's going to inherit both of these, and here's what it looks like. If you've got the black coat with lots of melanin, you are the typical black lab that everybody loves. Okay? If you get a different mix of your coat and your melanin, the black coat and the red melanin, you are a chocolate lab. And if you get the light coat and the darker melanin, or the light coat and the red melanin, either way, you're going to be a yellow lab. And you know they're still expressing a lot of melanin. You can see it around his eyes, his nose. His eyes are brown. There's black around his lips right here. It's just the coat itself that's going to be the lighter color. Even the ears are a little bit darker here. But these genes, two genes working together to make one trait. And you can see the combinations that'll give you a black lab. They're all over the place. And so now, just to kind of give you a brain teaser, there's also a silver lab, which was just recently, AKC certified or whatever. And so maybe you could check that out, like what kind of genetics gives us the silver lab. But there you have coat color in dogs. Multiple genes accounting for one trait. Pleiotropy. This is a good one too. Pleiotropy, I don't know why, it always comes with sad stories. But pleiotropy is kind of a neat deal. Pleiotropy is one gene influencing multiple traits. One gene, many effects. And they normally use a disease in most textbooks that I've seen and taught out of. They pick a disease, like sickle cell disease, or in this example, Marfan syndrome. And then they say, because of that one change, here's an effect, here's an effect, here's an effect, here's an effect. One gene, multiple different effects. So some tall, thin athletes have what's called Marfan syndrome, a potentially fatal genetic disorder. And this guy here in your textbook's an example of one of those guys who had that genetic disorder. And what ended up happening was there's some connective tissue problems with this disease, and so they're real tall folks, and their joints are a little bit I don't know what the word would be, maybe their joints aren't as good as they should be because their connective tissue and some of the fibers they make aren't all that great, and those same fibers help give your arteries the right blend of strength and flexibility to handle the high pressure. And his aorta, which is the biggest artery that comes right out of the heart, because that connective tissue defect of the disease he had, Marfan syndrome, the aorta actually tore and ruptured during a basketball warm-up, and he died from basketball practice. And that was one result of the Marfan syndrome, but so was the height, and so was the joints, and so were all these other things. So one gene had many different effects. So that story is in your textbook if you want to check that out. So here's a hard question. Nature versus nurture. How much of you is you because your genetics dictate that it's going to be that way, and how much of you is you because of the environment in which you grew up in? And that is probably one of the hardest questions biologists are faced with, and the answers are pretty unclear. But we do know that the environment can actually affect your genetics, and it can affect it 1 or 2 generations down the line. So the environment affects the expression of many genes, which in turn affects your phenotype, and that can include the behavior. If you would really just do me a favor here, there's 2 pages in your book for 13.6. Read those 2 pages because it's crazy. how the genetics can be affected by the environment. Methylation of certain lengths of DNA or demethylation of them is pretty insane. And you should really give 13.6 a read. It's only going to take you a few minutes to read those 2 pages, and it's pretty amazing. So basically, your genotype, the genes you have plus the environment you live in, dictate your phenotype. What you're expressed, what you're like, what we see, what we hear, that kind of stuff. They work together. And so epigenetics, environmentally driven changes in gene expression patterns can be permanent and they can be heritable. Earlier in your book, one of the chapters we skipped, sadly actually, was an example that these guys who had survived a famine had actually changed their DNA while they were surviving that long period of starvation, and their grandchildren were able to better withstand famine because of the genes they had inherited. And that's in human beings. That's not in bacteria, that's in people. And so we can make permanent heritable changes in our population based on changes in our environment. And so some of these can be gene controls, like chromatin modifications or RNA interferences that'll actually act on DNA. And so a lot of environmental factors affect DNA methylation patterns, and we can enhance how much of a gene we express, or we can suppress how much of a gene we express. And that's what's going to help us adapt, even in really small ways, to our environment. So some environmental effects that we can see that are just really, really obvious because people are tricky. Alternative phenotypes in water fleas. Individual water fleas will adapt to the environmental differences in standing bodies of water by adjusting their gene expression, which is kind of amazing. And then we also have seasonal changes in coat color. Temperature changes and length of day affect the melanin production in some animals' skin and their fur. So here's your water fleas. This guy over here grew up in water that had a normal oxygen content, and so he is green. This guy grew up in water that was oxygen poor, and you and I use a big protein called hemoglobin. Hemoglobin binds to oxygen and is a big part of the reason why our blood is red. This guy here, because he grew up in an oxygen poor environment, actually turned on his genes to make hemoglobin. And he made hemoglobin. And that is why he's red, because he is full of hemoglobin to survive that low oxygen content water. Same species, different environments, different gene expression. And a cool picture too. I can't even get my family to pose that nice for pictures, and look at these guys. They just look great, little water fleas. So here's a tricky one. This is a snowshoe hare. Okay, his fur varies by the season, and you know how your dog is always shedding just a little bit. You pet your dog, and you always get a little handful of hair. It's kind of gross. These guys are the same way. Old hair falls out, new hair grows in. All right? As the old hair falls out and we transition into fall, the old brown hair starts to slowly fall out, and the new hair that's growing in and replacing it is lighter in color. And eventually, when winter comes, most all that brown hair is falling out, and most all white hair is grown in. Then in the spring, the white hair will start to fall out, and the brown hair will start to replace it. So it's not like as dramatic as like a duck molting. It's more like slow, gradual shedding and replacement with some different colored hair. And so that's kind of an amazing thing. But that's an adaptation to some seasonal changes, and that's pretty cool trait to see in a population. So some different environmental effects. If you took botany with me, you'll really be geeking out pretty hard on this. There's an effect of altitude on yarrow plants. That's right, yarrow, Achillea millefolium, everyone's favorite plant from botany. Genetically identical yarrow plants will grow to different heights at different altitudes. If you plant yarrow at a low altitude, it'll grow up into this big, tall, flowery thing. It's real pretty, and we can use it for landscaping plants. If you grow it on a high granite peak, it grows low to the ground, and it looks very different. The leaves are the same, but it grows lower to the ground. And that's, I don't know how, but it senses its altitude and it expresses that in its genes. And it's kind of cool because high altitude places are windy and cold, and growing low to the ground is a great strategy. So good for Yarrow. I think that's awesome. Another one, psychiatric disorders. The environment is a big factor for disorders that we've seen, like schizophrenia, bipolar disorder, depression, mood disorders, and future treatments for these disorders might involve modifying the epigenetic markers on someone's DNA. So again, like I said, if you've got to read one section, read that last section in your book, because there's a much more detailed description of this, and it's really almost too much to digest. It's so crazy how much your genetics, I almost want to say, interface with your environment. It's weird. Weird. So please take that 10 minutes to read that or so. 3.6, 13.6, that's what you want to read. Okay, complex variation in traits. Lots of our traits don't appear in distinct forms. That stuff we were talking about with Mendel, purple flowers, white flowers, they're purple or they're white. A lot of our traits don't work like that. Often our traits are the result of some complex genetic interactions and some complex environmental actions, and we put all those together to make us us, and it's super complicated. Which is why it drives me nuts when students like, hey, my dad has brown eyes and I have green eyes. How weird is that? And I'm like, you have no idea how weird that is. I mean, it's insane. But there you have it. Everyone thinks it's simple like pea plants. It's not. Continuous variation. Traits with a range of small differences that continue variously, or vary continuously rather, across a population. So the more factors that influence a trait, the more continuous we're going to see that distribution of the phenotype in the population. So short tandem repeats can expand or contract quickly compared to the rate of mutation. Short tandem repeats are the same repeating DNA code over and over and over and over and over again. And they could be 20 long or they could be 500 long. And we can expand or contract those to make big changes in our phenotype. And we can do it in like a generation. We really can. It's crazy. So those DNA changes from those short tandem repeats can be preserved as alleles that we pass on to our kids and our grandkids. And so that's a way that we can make changes quick, and we can get those changes to our kids quick. So even though you might not be surviving the famine that well, your kids and grandkids are better prepared for the famine, the next famine, because of the modified alleles you're going to pass on to them. And it's kind of hard to digest that we can make changes that fast, but we really can. Here's one reason it bothers me when kids are like, check out how do I have green eyes? Well, what's green? This is variation in eye color in humans. So is this green? Is this green? Is that green? What's blue? That blue. Top row from the left, we've got all these photos that are different melanin expressions, right? Lots of melanin, darker eyes. Less melanin, lighter eyes. The melanins are browns and reds. And as you blend brown and red melanins, you get different colors. Like this amber color, you can get little traces of purple in there. You can get greens and blues mixed together. It's not uncommon to see brown flakes, brown specks in a green eye. There's lots of different things controlling this, and there's more than one allele controlling this, and so it becomes a little more complicated than we'd like to think. So, continuous variations are something we can measure on what's called a bell curve. when our continuous phenotypes are divided into measurable categories and they're plotted as a bar chart, they form a bell-shaped curve. And what it does is it shows us the relative frequencies of phenotypes across a range of values within our population. Here's what this looks like. They took biology students at the University of Florida, just the guys, and they used rows of bleachers, which I thought was really smart, and they categorized them by height. 1 inch increments, so 63 inches tall, 64 inches tall, 65 inches tall, 66 inches tall, all the way to 77 inches. And what you get is a really nice bell-shaped curve on the average, and there's little exceptions. It's not perfect. This row has one more guy than it probably should. This row has about one less guy than it probably should. This guy down here's got his arms folded. He's kind of cranky because he's the shortest guy in class, but that's okay because he's important. He's the edge of our bell curve, right? So that's continuous variation. Teachers can do this with grades, honestly. That is A+, that is F, that is C. Your kids should be on a bell curve. And you can shift the bell curve to the right a little bit and have your average be a B. It's kind of nice. So, yeah, it's a bell curve. Here you have the same thing. Bell-shaped curve in height for humans. So this graph is this put into, Excel or some sort of graphing program. And you run your little best fit line, and it works the average for you. And there you are. Just perfect little bell-shaped curve. So, that is the end of chapter 13. Points to ponder. I might have spoiled this one for you. What conclusions might Mendel have made if he had chosen snapdragons instead of peas for his studies? And there are 4 possible blood types in the ABO system. How many different alleles are in the human population for that particular marker? Another good question. So that's the end of chapter 13. Ask me questions in class. We'll do some Punnett squares. It'll be a good time.